I'm Kirti Vasan, first time as a applied microbiology. We are discussing light reaction. First, what is light reaction? The light reaction take place in this grana of the chloroplast. Here, light energy get converted into the chemical energy as a ATP and NADPH. In this very light reaction, the addition of phosphate in the presence of light or synthesizing of ATP by cells known as a photophosphorylation. Photophosphorylation. Photophosphorylation is the process of utilizing light energy from photosynthesis to convert it to ADP to ATP. In this process, synthesizing energy-rich ATP molecules by transferring of phosphate group in ADP molecules in the presence of light energy. Photophosphorylation is offered two types. They are cyclic photophosphorylation, non-cyclic photophosphorylation. Cyclic photophosphorylation. The photophosphorylation is a process of which result into the movement of electrons in an cyclic manner for synthesizing of ATP molecules is called cyclic photophosphorylation. In this process, plant cells just accomplish the ADP to ATP for immediately energy for the cells. The process of usually take place in the thylakoid membrane and photosystem 1 and the chlorophyll P700. During the cyclic photophosphorylations, the electrons are transferred back to P700 instead moving into the NADP. From the electron acceptor, this downward movement to the electrons from the acceptor to P700 result in the formation of ATP molecules. This is the cycle of cyclic photophos photophosphorylation. The sun light source was uh, photons received P1 PSI1 P700. The electron will be lost excited to the primary acceptor. The primary acceptor was ferridoxine reduced substance. Next, the electron transporters involved to ferridoxine ferridoxine and cytochrome complex and plastocystine to involve to add the another electron in the same PS1 700. They are releasing two ATP molecules. Next, non-cyclic photophosphorylation. The photophosphorylation process of which result in the movement of electrons is a non-cyclic manner for synthesizing of ATP molecules using the energy from excited electrons provided by photosystem 2 is called non-cyclic phosphorylation. This process is referred to as a non-cyclic phosphorylation because loss of electrons by P68 P618 of photosynthesis 2 are occupied by P700 of the photosystem 1 and are not reverted to P608. Here the complete movement of the electrons is in a unidirectional or in a non-cyclic manner. During the non-cyclic photophosphorylation, the electrons released by P700 are carried by primary acceptors and are finally passed onto the NADP. Here, the electrons combine with the protons H plus, which produced by splitting up the water molecules and reduce the NADP and NADPH2. This is the cyclic or pathway. This is also called the Z pathway. Starting with PSI1, the light energy will be observed PSI1, PSI 680. The electrons will be excited in the FRS. FRS means ferridoxine reduction substance. The ferridoxine reduction substance was um, carry out electron carrier. The electron carrier involved in ferridoxine FDNADP reducer. Then this process is carry out electrons carrier. This process will be NADPH2 converted NADPH2 NADP converted into the NADPH2. The PSI1 was loss of one electron. To manage that, PSI2 was give one electron. PSI2 was exited state give to the electron will be PSI1. The PSA2 was a loss of electron. To manage that, involved in H2O involved in give electron and one photon, the releasing oxygen. This is the reference of light reaction and phosphorylation. Thank you.